Hi, my name is Thanadoran. Now I'm going to present a study of phylogenetic relationship of the giant pure millipede genus Siphonia gray from northeastern Thailand. This is my mini project that I have done under the supervision of Dr. Rasapon Sichonchai. Giant pure millipedes are the arthropods in class Dipopoda, order Spherotheria. They are decomposers in terrestrial ecosystem. They have 10 to 100 mm of body length. They have 13 segments, and they have 21 pairs of legs. And they are capable of assuming their body into a spherical shape. In males, the last one or two pairs of legs form theropods, which help them in reproduction. In Thailand, over 244 species of millipede were recorded, but only a few species of giant pure millipede were found and described, especially the one that is commonly found and yet lack a systematic study, genus Siphonia gray, which is recorded just 7 species, namely Siphonia lanaensis, Siphonia prain, Siphonia panhaai, Siphonia inhofe, Siphonia golovaci, Siphonia siamensis, and Siphonia viridisoma. And most of them are classified by using morphological character. For example, the surface and the target, the antenna, and the tail pod. However, some morphological character of some species seems to be similar and vague. For example, this is the theropod of Siphonia siamensis and Siphonia lautica. You can see that their shapes are somehow similar. Therefore, only morphological characters may not be able to show the distinct differences between these two species or some other species. And recently, DNA technologies were used widely in taxonomic study of the giant pure millipede to identify the species by using molecular data. And in addition, the molecular data is really helpful in the study of phylogenetic relationships of these giant pure millipedes. So we decided to use molecular data to answer the distinct differences between them. And here are our objectives. First, to study a phylogenetic relationships of the giant pure millipede genus Siphonia gray from northeastern Thailand. And second is to learn and practice research skills, for example, collecting samples, studying molecular data in the laboratory. And let's start with specimen collection and identification. We collect the specimen from 4 different limestone habitats in Khon Kaen province, and we also use the specimens from the previous collection from Tak province and Nakhon Nayok province. And after we take the live photo, we euthanize them and store them in 95% ethyl alcohol, and the specimens firstly were identified by using morphological characters, following the previous work from Westerner and Rikitakan. Then we choose the representative to apply the DNA technique in order to study their phylogenetic relationships by analyzing their molecular data. And the next step is DNA extraction step. We use 3 to 4 of their hind legs to apply the DNA extraction technique by using nucleospin tissue DNA extraction kit and its protocol. And these are the steps that I have learned and practiced in the protocol. For example, in order to make the enzymes and the buffer work effectively on the tissue of the arthropod, I have to cut their legs into a very small pieces, and also I have to mix the mixers and incubate them and centrifuge them following the protocol until I get the DNA extracts. After that, we will increase the number of the DNA by using polymerase chain reaction technique or PCR technique, targeting gene cytochrome oxidase subunit 1 or CO1 with 658 base pairs. And this technique will be done in the PCR machine with the condition adapted from Westerner 2015. The materials are DNA extracts, LCO forward primer, SCO reverse primer, master mix, and DDH2O. Firstly, we will apply the PCR technique in the small volume, which is 12 microliters, and use PCR gradient technique in order to find suitable condition for the DNA of our specimen. And here is the detail of the condition that we adapt from the previous work. And you can see in this red circle, this is where we apply the PCR gradient technique, which is that we vary the temperature between 12 columns in this PCR machine. Then we will check the result by using gel electrophoresis technique, which is that we prepare the PCR result into the gel and check it under the UV light. And as you can see, at the edge of the gel, there is the DNA ladder showing the positive result, and between them there are 12 columns that we vary the temperature, and the results show that all of them were suitable or beautifully positive. So we choose 50 degrees Celsius. And after we know the suitable condition, we will apply the PCR technique again, but this time with the large volume, which is 60 microliters. And here is the detail of the condition that we adapt. You can see in this red circle, we change from the various temperature to fixed temperature, which is 50 degrees. And then we will check the result again, and the result went beautifully. After that, we send the large volume result to the bionics company in South Korea for DNA sequencing. Then they send us back the files containing sequence data of our specimens. Then we will insert the files into Mega 7 program, which is the program that we edit and align the sequences. 17 sequences of our specimen were analyzed together with those of the related groups from GenBank, which is H. And we use these sequence data to construct the phylogenetic trees by using maximum likelihood statistic method, with the number of replication bootstrap equal to 100. And here is the result. This is the phylogenetic tree, and those with the highlights are the specimens of our collection, and those without highlights is the database from GenBank. And let's look into the detail. First, start with the left one. On the top half of the tree, this is from genus Siphonia, and the lower half of the tree is from genus Sphaerobellum verhoff, which is the outgroup of our study. And let's move to the right. You can see that each clade represents each species, and some of the specimens are the recorded species, which are Siphonia prain, Siphonia inhoffi, and Siphonia golowachi. But there are two groups that form their own clades, separated from the recorded species. And due to the morphological and molecular data, we assume that they are the unknown species. The first one with the blue highlight, we name it the Siphonia species A, and the second one with the purple highlight, we name it Siphonia species B. And in terms of diversity of the Siphonia gray in Thailand, there are 3 species that were collected from northeastern Thailand, which are Siphonia inhoffi from Khon Kaen and Loi province, and Siphonia golowachi from Nakhon Ratchasima and Nakhon Nayok province, and the last one is Siphonia species A from Khon Kaen province. And in Thailand, there are 6 species with sequence data, including Siphonia species B from Tak, plus 3 species without sequence data, which means there are 9 species of the genus Siphonia gray in Thailand. And in terms of phylogenetic relationship, the northeastern group highlighted with green color is the monophyletic group, which means they form their own clade separated from other groups. And as you can see in this red circle, the northeastern group came from the same clade with the Siphonia species B from Tak province. This means that the northeastern group is related to the Siphonia species B, and also this another red circle. The Siphonia species B and the northeastern group came from the same clade with other species from Thailand and its neighboring countries. This also means that they are related, but not as much as the previous one do. And our discussion is that this study supports and is consistent with the previous taxonomic research of the giant pale millipede genus Siphonia gray. For example, it supports the previous record of the new species of the giant pale millipede genus Siphonia gray. And here is our conclusion of this study. Regarding the previous study and this study, there are 3 species of giant pale millipede genus Siphonia gray in northeastern Thailand, including the unknown species A from Khon Kaen province. And in Thailand, there are 9 species, including the unknown species B from Tak province. And our phylogenetic study reveals that the northeastern group is closely related to the Siphonia species B from Tak province, and these two are related to other species from Thailand and its neighboring country. And this study supports and is consistent with the previous taxonomic research of the giant pale millipede genus Siphonia gray. This study also shows that using both morphological and genetic data is more reliable and can be used to identify species in the taxonomic grouping. However, this is a preliminary study of sequence data, and many of them have not been studied. Also, some morphological characters haven't been fully compared. Therefore, we still have to wait for further study. Acknowledgement, IPST, DPST, and Department of Biology, Faculty of Science, Khon Kaen University. And finally, thank you for listening.